As she said, I'm Brooke, and I'm here to round out the trio of Williams Lab presentations. And Jay just talked about the past evolution of the ribosome. I'll be talking about taking the ribosome and evolving it further. And to do this, I'll be using the Saccharomyces cerevisiae mitochondrial ribosome as an orthogonal evolvable translation system. So let's just start with the translation system. As Jay briefly touched on it, it is a system of a lot of interconnected parts. You have mRNA, tRNA, amino-acyl tRNA transferases, and at the core of it all is the ribosome. And through billions of years of evolution, this ribosome has evolved to take the alpha L amino acids, make amide bonds, and we've got these peptides, proteins coming out of it. But at its core, it's not just making these amide bonds. It is a polymerization machine. It's finely tuned in the way life is right now, but it is making polymers. So that means its function can be expanded to make other kinds of polymers. And that's actually of a lot of interest when you use like non-canonical amino acids, and other things because it's so efficient at this. The problem with evolving the ribosome is that there's a really large sequence design space. We've got the ribosomal proteins, we have the ribosomal RNA. It's just huge. It's not possible to do in vitro evolving of the entire ribosome. You can't touch it. So you have to go for an in vivo approach that's more high throughput when you have it in cells. It's easier to assay. But because the ribosome is so central, once you start touching it in vivo, the cell dies because almost all mutations are deleterious and it's so essential for survival. So that's when we move on to an orthogonal translation system. So here is an orthogonal translation system in E. coli. You maintain your native translation system for all the native processes, so you still have your proteins, and then you have another ribosome operating at the same time, and this is the orthogonal one. And you have your mRNAs directed to which ribosome they're going to go to, so you have your native mRNA and your orthogonal mRNA. But there are a few problems with this system in E. coli right now. So as you can see, there's this like red tether between these two, the large subunit and the small subunit, and that's to prevent Franken ribosomes, because how do these large subunits know which small subunit to go to? So that's a big issue. The other issue is it's not just RNA that makes up ribosomes. As I said before, you've got your ribosomal proteins. And as the cells divide and make more ribosomes, they don't know where the protein should go to, which ribosomes they should go to. So they can't, using this orthogonal system, you can't touch the ribosomal proteins. You can only touch the rRNA. And even with that, they can't touch the peptidyl-transferase center. So it's a very limited system, which is why we thought the Saccharomyces cerevisiae mitochondrial ribosome would make the perfect orthogonal evolvable translation system. So Saccharomyces cerevisiae are a type of budding yeast. And as eukaryotes, they have cytoplasmic ribosomes and ribosomes in their mitochondria. And these mitochondrial ribosomes have their RNA mitochondrial encoded, but their proteins in the nucleus. And they're completely separate. So their translation doesn't affect each other. They, while the ribosomal proteins are nuclear encoded for the mitochondria, they have a specific sequence sending them to mitochondria. So there's no issue of like cross-talking of these two. And so I have the system for using these mitochondrial ribosomes as my system, I guess. So we start with a perturbation of the mitochondrial ribosome. It could be editing the mitochondrial ribosomal proteins. It could be some other kind of stopping of mitoribosome function. And when I do this, there are two possible outcomes. Either I have functional mitoribosomes or non-functional mitoribosomes. So I can test that using a different type of carbon source in the growth medium. In this case, it's glycerol for these cells. It's a non-fermentable carbon source. So they need mitochondria functional to grow on this non-fermentable carbon source. So we have functional mutations, non-functional mutations. But this isn't where the story ends. And this is what's special about Saccharomyces cerevisiae. They do not need functional mitochondria to stay alive like we do. So just because the mutations are non-functional doesn't mean that the cells are dead. So I can take these cells with the non-functional mitoribosomes and then continue to grow them in a non-selective medium, so using glucose as the carbon source. And this allows them to accumulate spontaneous mutations as cells divide, and then I can test to see if any of these mutations are compensatory to allow the mitoribosomes to function again. And I do this by once again going back to the glycerol as the carbon source. So to test my system, I first started with ML50 as a mitoribosomal protein. Here we have the mitoribosome, and then in cyan we have this ML50, and I truncated it. So I cut off that part in red. And this is a mitochondrial specific protein, so we don't see it in our cytoplasmic ribosomes or anything like that. And I found with this truncation that all these cells are actually YPG positive. So they can grow in the non-fermentable carbon source, which means their mitoribosomes are functional. But that gives us a very limited view. So I have this strain of yeast with GFP that's encoded on the mitochondrial genome, so I can use this as a readout of mitoribosome function. And that's what I did here. So in red, we have a type of wild type yeast. So it's a non-fluorescent strain. In blue, we have the positive control, so it's a fluorescent strain. And then in orange, we have the truncated protein. And I grew these and ran them through the flow cytometer, which measures size and fluorescence per cell. So this is all on the single cell level. And here we have some flow cytometer results. So this is average fluorescence per cell in relative fluorescent units. And then these are the two media types. So using YPD, which is glucose, and then YPG, which is the glycerol. And once again, the glycerol means we have to have functional mitochondria. And with the non-fluorescent strain, we have low levels of fluorescence. And then with the no truncation, we have a high level of fluorescence, and then a significant decrease in fluorescence with the truncation. So this shows that even this mitoribosomal truncation, while we still have function, we have a decrease in function. And then on the right here, we have this histogram showing in the same colors the peak shifts with the different types of ribosomes. And then, as I said earlier, we were talking about perturbations. So I can easily touch these proteins, but I can't touch the rRNA because it is mitochondrial encoded. But there's another way I can perturb the rRNA. So mitoribosomes are actually more similar to bacterial ribosomes than like their cytoplasmic counterparts. So you can use antibiotics to selectively target mitoribosomes that you can't without touching the cytoplasmic ribosomes. And I started with chloramphenicol. So chloramphenicol prevents peptidyl transfer, usually in bacteria. So this is an E. coli ribosome right here, in the peptidyl transferase center. And then it is known that mitoribosomes, particularly Saccharomyces cerevisiae mitoribosomes, can accumulate one single nucleotide polymorphism to have chloramphenicol resistance. So I went through this evolution experiment using my workflow I showed before, and here we have some suppressor mutants I was able to get. So we have wild type up here, and that was the original strain, and then down here we have 5 suppressor strains. And then I sequenced their 21S, found the single nucleotide polymorphism, and then used that strain in the flow cytometer to see what happens to mitoribosome function. So we once again have this bar chart on the left with the fluorescence per cell, and we now have added, instead of just the YPD and YPG, chloramphenicol to the mix. So with just YPG, we have very similar levels of fluorescence between the positive strain and the chloramphenicol resistant strain. And then once we move to YPG plus chloramphenicol, only the chloramphenicol resistant strain is fluorescent, and it has a very similar level of fluorescence. So what we can say here is that the chloramphenicol resistance does not inhibit mitoribosome function, even though there is a mutation in the 21S of the mitoribosome. And here is the histogram with GFP fluorescence on the bottom, showing the peak shifts when we have chloramphenicol in the YPG. And with that, I would like to share my conclusions that the S. cerevisiae mitoribosome function decreases with the short truncation of ML50. A compensatory mutation in the 21S of the mitoribosome gives resistance to chloramphenicol without hindering function. And lastly, and maybe most importantly, the S. cerevisiae mitoribosome is an isolated orthogonal evolvable translation system that we can then use to do further perturbations of the ribosome. And I would like to thank Lauren Williams, my PI, and Kirill Lobachev and Wen Ying, because they've been so instrumental in getting me started in yeast. And then lastly, Professor Martin Ott from the University of Stockholm for kindly gifting me this strain of yeast.
Great. Do we have any questions?
So I think this is probably a pretty simple question because I don't have much background in this stuff. But how does the cell keep living once the mitoribosome doesn't function anymore? What replaces its function?
So as long as you're growing it on glucose, you don't need functional mitochondria.
Okay.
So they just don't use them at all.
So does the glucose somehow replace that function?
Yeah.
Okay.
Yeah.
That was my question. Thank you.
Thanks.
Yes.
So did you say you were going to be evolving them in response to protein mutation as well? Like the
Yeah, so that's in progress right now.
Yeah. So do you have any intuition as to the number, as to how many generations that might take? In Lauren Williams' lab where we do the evolution experiments, and I'd love to talk to you about that sometime.
Yeah, so we've done some successfully with truncations where I don't measure like number of generations, but we're talking about like growing in liquid overnight, plating on YPD for a few days, and then replica plating to YPG. So we're not talking about long periods of time here.
So they're fixing themselves in a reasonably short period of time from these perturbations.
Yeah, and the chloramphenicol resistant ones were actually really fast.
Okay, so these are high. So you're actually finding highly accessible evolution.
Yeah, I'm sure the like larger perturbations we do, we might have to add like a chemical mutagen, but yeah.
But still, so it's working fast and well. That's great.
Yeah.
I love it.
And do we have any other questions or anything from online? Great, we can move on then. Thank you.